many of the compounds are being, some medicinal chemist will do all the kind of reactions, but slowly it will narrow down. And see the last graph, the process chemistry duration and the medicinal chemistry duration. That means medicinal chemistry will try our small. It is easy to handle hazardous things in the fume hood. Butyl lithium. Have you heard of butyl lithium? Any strong base? So these strong bases you can use. But in the large scale, you got a compound which is very active. After you get a compound, you have to synthesize at a larger doses. So you have to give it to the animals, maybe higher animals, and then other preclinical studies, and then clinical studies. So how will you synthesize if the route of synthesis is not giving you the product in a good yield, as well as it is liberating some hazardous material. I am giving you one case study example. When I was working in Dr. Reddy's, there was one compound of DRL. This is Dr. Reddy's laboratories. So there I was working on anti-infective compound. It was 11 steps. Okay, and luckily the molecule was also active. So my lab in charge told, okay, now we have to give this molecule to the process chemist, because now they are going to test on the animals, right? So 500 and with 99% purity. It's difficult to get 99 point, pure, more than 99 it should be there. So there was one step actually which was killer. So one step, we used to have sodium hydride. Sodium hydride is the base, and sodium hydride, if you take, so what happened actually, my team, we were there working in a very lower level as a laborer. So we went to the process chemist people. We told them, okay, this is the protocol. We have to handle. We have to give them, deliver them that this is the work. Then they will work at a higher level. When we gave them, okay, sodium hydride, it didn't work. What happened? It caught fire. So then they said, no, we cannot handle sodium hydride because quenching is also difficult. Once the reaction is over, we have to quench it and it will catch fire. So then they said, give us a substitute. So we tried, we did a lot of methodology changes, and we finally got the sodium carbonate, which is an organic base, easy to handle, water soluble. So it worked. But the problem was on TLC, the starting and the product are the same RF. So we gave the sodium hydride reaction went like in 6 hours, but sodium carbonate went in 24 hours. They said it's okay. We can keep the reaction for 24 hours, but we cannot handle sodium hydride, which is costly also, difficult to handle, and sodium carbonate is easy. So that's the way process chemistry drives the outcome of the lead molecule. Okay. So this was just example to talk about. Then limitations, we have to establish reactions each and every time, and each step has the limitations. So we cannot satisfy all the principles of green chemistry. We have to see, like in medicine, like in pharmacology, you go for the benefit to risk ratio. Likewise, we also see, okay, how many steps? What is the yield? If the effluent is there, whether it is dangerous, toxic or not, like that we compromise at some level. No reaction is completely green. Okay, no reaction is completely green. We call green chemistry, but it is nothing exactly green chemistry. Yes, we try to use some solvents which are not hazardous in nature. You all biologists use DMSO, am I right? For screening. Now, believe me or not, DMS is also going to replace by some other solvent. US people are pushing hard that DMSO is also not good solvent, so we will replace it with some other solvent. Okay, so DCM, benzene is also gone, already gone. So many solvents you will see they will be gone now. So importance of so we have to write down all the protocols, which solvent, how. We publish a paper, but we do not disclose it completely. I remember, and you will be also facing some of the chemists. You want to synthesize something, but reaction is not going. Right? What could be the reason? Either you are doing something wrong, or the report is not correct, or some trickiness is there. Okay, so sometimes it is not mentioned, you have to use dry solvent. Okay, so you have to repeat it 3-4 times, then conclude, okay, I am wrong or the data reporting is wrong. Therefore, I used to follow organic collective volumes, which are never wrong, because it has been checked by third party, and third professor will be standing and he will be seeing, okay, what is happening in the reaction. Okay, if the reaction is reported in organic collective volumes, please go through that. That is not challenging. Standardized, well standardized. Okay. What is going on now? What if I tell you, you have some molecule, like any drug you talk about, camptothecin, allotinib, any drug you talk about. What if I say there is drug? Now I want to make, this is without nitro, I want to make a nitro direct. Or I want to put some benzene here. What will you do? Any idea? You will change the route of synthesis. You will change the starting material. If it is the 11-step synthesis, you will start with a substrate where already phenyl ring is there. Are you getting my point? Again, I am this, saying it. If you have a drug which has some problem, now you want to develop its structure-activity relationship, or it has some problem, you want to modify it. What will you do? You want to put some benzene here or some other group here. What will you do? If the simple reaction is not possible, if you have a benzene ring and you have a hydrogens, you want to replace that. What if, जैसे हम बचपन में क्या करते थे, अगर ये हाइड्रोजन की जगह पे ये होता तो क्या? Think there is no problem in thinking, right? So the thought, scientists thought that if I replace one of the hydrogen with some other phenyl, it's possible. Where reaction looks very difficult, chemistry looks very difficult. There is no, hard known chemistry known. Okay, so that is called CH activation. That also comes in late stage functionalization. That means you have a drug. Now you want to modify. You want to modify. You want to make a new drug. If you are not aware of this CH activation or late stage functionalization, this is called late stage functional LSF. What you will do? You will start from scratch. First step, then second step, or you will be looking for some commercial reagents. Okay, whether this with this is available or not, with fluoro available or not, trifluoromethyl available or not. So this is actually this also is not a new concept. It started 20 years back, but it is taking a boom now. CH activation. So you have a drug which is having a pyridine, benzene, whatever, and you have a hydrogens which are difficult to remove. A base cannot remove that. Okay, it's difficult to remove, but you are now smart. You have a good chemistry. You can replace that hydrogen, which is inactive kind of hydrogen, with a new compound and put it like, okay, you are sitting down. It is a new drug now. It's a new chemical entity. You have to do all the parameters again. And you can have a better or less activity. It may be having an improved pharmacokinetic profile. So that is CH activation. I know it's a costly affair. Chemists have worked day and night for this CH activation, but it has really worked at the end. So this is the example. It has been shown that this is the drug. For example, this drug can be modified. A fluorine ring can be put any of this carbon. This is sp3 carbon, and or any of this carbon, which is sp2 carbon, benzene ring. That means you can replace hydrogen of a benzene and put a fluorine group, which is very difficult. It's very difficult. It is not an easy chemistry. It is not a textbook chemistry, I would say. And this putting fluoro here is also difficult. So this can be possible with a concept called CH activation and late stage functionalization. CH activation is one of the technique can be used in late stage functionalization. Okay, so it can be used in macromolecular chemistry also. Like you might have heard of conjugates, DNA conjugates, RNA conjugates. There also it is visible. That's why both of them they got Nobel Prize. That they just put an azyl group and made a click triazole ring, and they got Nobel Prize. Sharpless and this lady, both of them they got Nobel Prize. Sharpless got 2 Nobel Prize rather. Okay, so this is you have a biomolecule. Think about you have a protein which has SH group free. How it is possible? Any student? SH group. Cysteine, which is amino acid. But cysteine generally in the redox reaction, cysteine is time, because sulfur bond formation is frequently happening. So you have a free SH, and using this palladium chemistry, you can make S-aryl group, which is this should be water soluble because bioconjugate chemistry works in water, not in solvent, which we do in the lab reactions. So you have to develop the reactions. Now, because I am a consultant to one company, they are working on diagnostic agents. Trans-Asia Biomedical. There also we have developed some compounds where like in the water acid A B chemistry just simply add the things because you want to see the color, you want to see the chemiluminescence. There you don't the patient is not the MSc guy is not bothered about the mechanism of reaction. He or she is bothered about what is the end result. So it should be easy and quick chemistry. Okay, that's why that the other reaction is called click chemistry, because it clicks like triazole bond formation. So this is also possible. You can modify a macromolecule. Okay. Then these are some drugs which are having emerging building blocks of medicine. When we wrote that perspective, 5 years after we approved drugs, we found that many of the drugs now having boronals. So as a teacher or as a speaker, I would request all of you go and read how boron compounds are useful in, as a drugs, how they are metabolized. Okay, are they toxic? Why they are replaced? Why this boron is coming into picture? Their approved drug, FDA approved drug, which is having a boron. And you can see that you can generate a boronic acid derivative by simple Suzuki coupling, which is difficult to, in the outside reactions. So you can develop a reaction. Okay. Biocatalyst, we discussed in drug discovery, you can use some enzyme, you can use some enantiomeric chiral compounds because enzymes are chiral in nature, right? So they will selectively catalyze once reaction in pseudoselective or pseudospecific manner. So this is another kind of reaction. See the last reaction where you have a carbonyl and this is the carbonyl, and here in the presence of enzyme, and this is the actually racemic trans and cis. Okay, so one of the isomer is being reacted with this, other is left aside. Okay, so they are so specific that only one compound is reacted, other is not. So having this knowledge, you can do this reaction in the using particular this enzyme and all this system. These are all user-friendly reaction, I would say. Okay, and again the C-N bond formation reaction. Have you heard of C-N bond formation reaction? Not cyanide, C-N bond formation reaction. So which one Hartwig reaction is more popular? Go and read. This is today's reaction, or means in the industry people are still using it, C-N bond formation reaction. Okay, and this C-N bond formation reaction is nothing but C double bond ONH reaction. Here no short-term common reaction is being used. Okay, so it is first converted into coenzyme and then it is being, because this carbonyl is activated. Now NH2 can attack and it can make a amide bond and this enzyme is free. So bacteria catalyzed, virus catalyzed, we can use bacteria machinery. Bacteria is smart, virus is smart, they are using our machinery. Why not we can use their machinery to get like the same reaction for attacking them? Okay. So I talked about one more thing, late stage functionalization. So you have a drug, at the end, you don't want to synthesize all the steps. I'm just, because, for example, you have a drug which is having a higher metabolism, clearance is high. Okay, a drug is there, benzene ring, there is a carbon, that's benzene ring is free. So you want to put a fluorine. You have heard of isosters, bioisosters, right? Bioisosterism. So if you replace methyl with fluorine, the metabolism is slowed down because methyl otherwise gets converted into carboxylic acid and then decarboxylated and activity is lost. So if you replace methyl with fluorine, it can prevent its metabolism in the liver, and the plasma concentration will be sustained for longer time. So now you have a compound, activity is very good, you have a methyl, now you want to replace with methyl, fluorine. What will you do? You will again synthesize a compound with fluorine. So many steps. What if you find out a mechanism that in one go, you have a drug, just one reaction, you replace methyl with fluorine or something, whatever you want. That is late stage functionalization. You have the drug, at the end you are modifying it, okay, for any of the reasons, like to improve the kinetics or dynamics. Okay, so this is a major evaluation of organic synthesis. So earlier we used to have simple +1 + 2 textbook chemistry. If you remember, functional group, cyano को COH में बदल दिया, इसको उसमें बदल दिया, all the conversion. The first thing. Then metal catalyzed came into picture, palladium and all this thing. Now people are not using much palladium. Rhodium, ruthenium, these are all heavy metals, and heavy metals तो हैं ही, they have some other problems. Some are, some catches fire at a larger scale. I remember I was working with Reni nickel, बड़े scale पे आग लग गई. So you cannot handle the bigger, things. And palladium also has the same issue. So now people are working on 3D other elements like iron, copper. Copper chemistry is now very popular. Iron chemistry is popular. Easy to handle, easy to recover also, easy to reuse also. Okay. So CH bond activation is also a kind of functionalization. New disconnection. Have you heard of disconnection approach? Retrosynthesis. MSc chemistry कौन से हाथ खड़े करो? M from chemistry. Disconnection means, for example, if you want to make this auditorium, what will you do? जो architect planning करता है, it is nothing but disconnection. Architect पहले ये बनाता है, वो बनाता है. Like that, we also do architecture kind of thing for the synthesis of target molecule. We design in our mind, okay, this should be the target molecule, ऐसा structure होना चाहिए. Negative product छोड़ दो. ये होना चाहिए. So what we'll do? We'll start building it or disconnecting it. कैंची करेंगे. Disconnection approach. So okay, we will disconnect. We will have plus minus this thing, and we will try to make a bigger molecule. And utilizing the known E.J. Corey was the first one who gave this disconnection approach. He's a very famous person. So this is the late stage functionation. At the end, it's a very simple picture that you have a drug. Capsule की form में दिखाया. You have a drug, and it has some hydrogens. How many hydrogens? 1, 2, 3. But simplified pictorial presentation. That it has 4 hydrogens, but we are selectively replacing one of the hydrogens with some group, dark group, anything. You know, this is very difficult to remove these hydrogens by simple known textbook chemistry. So that's why it has a different feeling, late stage functionalization. Okay, so LSF in drug discovery, you can see that various methods are available where functional groups can be put on at the end of the drug, any approved drug, and structurally complex molecules you can use. Anything, even I told that you can modify the macromolecule also. And functional group tolerance should be there actually. And high chemoselectivity and size. Chemoselectivity means like, and regioselectivity also, like I told there, other functional groups should not be disturbed. Which you want should happen. Which you want, like, and regioselectivity means there are 4, 5 hydrogens. But you want only one hydrogen to be replaced. This is called regioselective. Type specificity. Okay. And no specific groups. Free कोई और group नहीं लगाना. अगर protection करके कर रहे हो, फिर ये chemistry late stage functionation नहीं है. Okay, that will be classical chemistry. One step में होना चाहिए. And small subsequent installations. आप थोड़ा बहुत install कर सकते हो groups को, but major नहीं. Opportunities क्या हैं? क्यों करना चाहते हो? SAR develop करने के लिए. You know SAR? ये तो पता होगा. Structure activity relationship. मेटाबोलाइट प्रोटीन मैंने बोला कि आपका सिंपल बेंजीन है या सिंपल pyridine है, वो जल्दी क्लियरेंस हो जाएगी उसकी, मेटाबोलाइट खराब होगी. तो you can put some hard group fluorine which will retain the activity but improve the pharmacokinetics. So metabolic production, chemical biology tool जिसमें नोबेल प्राइज मिला, क्लिक केमिस्ट्री है सब. फिजिको केमिकल प्रॉपर्टीज अल्टरेशन, नेचुरली आप कुछ ग्रुप लगा रहे हो, नाइट्रो कुछ भी और लगा रहे हो, बेंजीन एक और लगा रहे हो, तो व्हाट विल हैपन? लिपोफिलिसिटी बढ़ भी सकती है. पोलर ग्रुप लगा सकते हो तो उससे लिपोफिलिसिटी डिक्रीज होगी, फिजिकल इंप्रूव हो सकती है. हैंडल फॉर फर्दर बायोकंजुगेशंस, जैसे अभी हमने बोला सल्फर को you can make a peptide bond or you can conjugate. People are now using alkyne chemistry, alkene or alkyne conjugates with the this thing. How these antibodies are being monoclonal antibodies and you know the what is this ADCs? Have you heard of ADCs? Antibody drug conjugates. Antibody drug conjugate, that's a real conjugate actually. Medicinal chemist has a role to play, but biologists, pharmacology people, they are using ADC. So what is ADC? ADC, you have a drug which is called payload or cytotoxic material. It could be any cancerous, anti-cancerous compound. You have a linker, and at the end you have a succinimide or some group. So you have a paired protein which has so many SH free or NH2 free amino acid or acid side chain amino acid की बात कर रहा हूँ. बीच वाला तो पेप्टाइड में बनाया है. Side chain SH cysteine, side chain NH2, arginine type, guanidine. So they can now conjugate with the drug. So what will happen? A big drug is there, big macromolecule is there, protein. उसके ऊपर बहुत सारी drugs लगी. It is also a kind of Professor Bansal को मैं कहना चाहूँगा, दोनों बंसल सर को, कि it is a kind of drug delivery system actually. That a one monoclonal antibody is carrying so many drugs. Because it's a big molecule, right? So, and it has an advantage because monoclonal antibody is acting from the surface and it is delivering the drug and it is penetrating and so many molecules are being delivered at one go. Okay, so it's a very good. ADCs are approved. We FDA approved drugs. Protecs are also coming up. This is another technique, another method. This is also chemistry, a chemistry-driven project. Protecs, proteolysis targeting thymidase. You have the drug, you put a linker, okay, and you have a drug and you put a linker, you put E3 ligand, which is also a small molecule. What will happen? It will go into the cell, it will utilize ubiquitination process and the degradation will take place. किसका degradation अभी सर ने western दिखाया था आपको. So western blot से you can check, okay, this protein is being degraded. So concept is going from inhibition to degradation. Okay, so every technology, every method, every modality has a pros and cons. Of course, proteins are also suffering, but they are going into picture, particularly in oncology and neurodegenerative diseases. Because neurodegeneration may be वो protein degradation कर रहे हैं. They want it. So these are the opportunities and role of LSF मैंने आपको बता ही दिया है. I don't want to this thing. So these are some examples where drug has been modified. This is, you can go to this Nature reviews. It's a very good journal, and all the success stories are being published in this. And now this could be two types. CH activation could be of two types. Either you can direct it or you can do innate. Innate मतलब inherent properties. So directed means मैं पढ़ के नहीं बताऊंगा. You see this drug. This is helicoxib, well-known drug. Celecoxib इतनी पुरानी drug है. It has one hydrogen this side, this hydrogen, this hydrogen, this. चार hydrogen बचे हैं ना इसके 1, 2, 3, 4. You can replace this hydrogen with iodine. अगर आप simple iodine chemistry करते हो, अगर chemistry के check कर लें textbooks में से, you need a double bond to put it. This double bond is not simple double bond. It is a benzene double bond. The electrons देते नहीं किसी को. Very difficult to remove this hydrogen. So this is not activated, but this hydrogen is being activated by this sulfonamide group. So this is directed. This is directed CH functionalization. You can utilize this कि उसके अंदर हमने एक group रखा है, which through this chemistry, palladium acetate. It is kind of, not kind of, it is actually आपने एक coupling reaction पढ़ी होंगी, Suzuki kite coupling. ये वही reaction है जिसमें हमने iodine लगा दिया. So what? इस iodine को आप replace कर सकते हो. Now it is a halogen. This halogen can be substituted by various SNAR कर सकते हो आप. किसी भी group से replace कर सकते हो. This is the late stage functionation, which is directed by some group. लेकिन दूसरा मेथड क्या है? Innate. Innate का मतलब है ये आपकी कौन सी ड्रग है? कोई पढ़ के बताना. ये दिखाई नहीं दे रहा. Loratadine, I guess. Loratadine. You can see this drug, and this has also 1, 2, 3. Deliberately, this hydrogen is being like colored red. So to show you, right? So there are 1, 2, 3. और इधर भी है देखो hydrogen. This side, है ना? यहाँ भी hydrogen. But particularly this hydrogen can be removed by it can maybe a salt like triphenyl phosphine salt, and this carbon अब इससे आगे तो कुछ भी बना सकते हो. एक्टिवेट कर दिया हमने, हाइड्रोजन रिमूव कर दिया और सिलेक्टिवली रिड्यूस सिलेक्टिवली. इतने सारे हाइड्रोजन थे बट ओनली वन. अब इसमें इनेट का मतलब क्या है? इनहेरेंट. इनहेरेंट मतलब हमने कुछ भी ग्रुप नहीं डाला इसमें. नो ग्रुप हैज बीन एडेड. But the question is, it is based on electronic and stirring factor. If you add acetic anhydride in this or triplylic anhydride, this nitrogen will make a nitrogen के साथ attack होगा और triplet के साथ ये NCOCF3 बन जाएगा, जिसकी वजह से इस कार्बन पे इलेक्ट्रॉन डेंसिटी बहुत कम हो जाएगी. It will be इलेक्ट्रॉन विड्रॉइंग ग्रुप की तरह इलेक्ट्रॉन सकर का काम करेगी. Because of that, this carbon will become electron deficient, and now you can put anything. कोई भी electron rich species. Triphenyl phosphine वैसे इतना ज़्यादा अच्छा नहीं है, but phosphorus has a lone pair of electron. It can attack here और nitrogen पे. उसके बाद ये salt बन गया. इस salt को आप use कर सकते हो किसी भी purpose. So selectively you can replace. You can do. ये salt water soluble भी होते हैं. You can make water soluble things also. Okay. So this is another example where this is innate. That already the drug has this property where we can utilize this property, steric or electronic factors to do the late stage functionalization. So these are the advantages and disadvantages. Like key benefits I have already discussed. So this thing and yes, step star सबसे बड़ी बात है cost effective. Last stage पे check करना है. फिर lead optimization, metabolics and bioconjugation ये हम discuss कर चुके हैं. Then emerging strategies and future directions. मॉडर्न अप्रोचेस में अब क्या चल रहा है ये भी तो इंपॉर्टेंट है ना. लेट स्टेज फंक्शनल चल रही है, इसमें क्या क्या और चल रहा है. तो यू हैव अ रेडिकल केमिस्ट्री इन फोटोरेडॉक्स कैटालिसिस, साइड सेलेक्टिव मॉडिफिकेशन यूजिंग लाइट. एलईडी पे बहुत काम चल रहा है लाइट एमिटिंग डायोड का यूज करके. एलईडी लाइट, ब्लू लाइट पर्टिकुलरली कि आप ऐसे रिएक्शंस को ब्लू लाइट दिखाओ. इवन आई हैव सीन सम स्टडीज वेयर सेल्स को ब्लू लाइट में रखा गया और उसकी रिएक्शंस का पाथवे चेंज हो गया. जो प्रोडक्ट बन रहे थे कुछ और बनना शुरू. radical chemistry you can use. Then biochemistry, electrosynthesis, sustainable functionation method. New modality I have already discussed. Now, 3 new modalities बहुत चल रही है मैं आपको बताऊं. ADCs, antibody drug conjugate, जिसमें chemistry का रोल है. Second है प्रोटेक्स, थर्ड है मैक्रोसाइकिल. सर आप सबको आप हमें पढ़ाते थे, बस मुझे सबने पढ़ाया, मुझे डॉक्टर साहब ने पढ़ाया. हमें पढ़ाते थे रिथ्रोमाइसिन, रिफैम्पिसिन, देयर मैक्रोलाइड एंटीबायोटिक्स. रिथ्रोमाइसिन इज अ मैक्रोलाइड, है ना, आपने पढ़ा होगा. अब उसी कॉन्सेप्ट को यूज करते हुए मैक्रोसाइक्लिक ड्रग्स बननी शुरू हुई. पर्टिकुलरली अब आप सोचो जरा, व्हाई पेप्टाइड्स आर नॉट यूज्ड एज ओरल ड्रग्स. पेप्टाइड्स आर नॉट सिंपल पेप्टाइड्स, बायोइंडिकेशन, राइट? क्यों? हाइड्रोलाइसिस हो जाती है उनकी, एमाइड एमिडेजेस हैं, सब पेप्टाइडेजेस हैं, सब खत्म हो जाता है उनको. एफर्ट बहुत है. सोचो जरा अगर एन टर्मिनल और सी टर्मिनल को ऐसे कर दिया जाए, समझ गए? तो वो साइक्लिक बन जाएगा और बीच में से कुछ कुछ अमीनो एसिड्स को अननेचुरल अमीनो एसिड कर दिया जाएगा. वो अमाइड दिखेगा ही नहीं उसे. या नाइट्रोजन पे हाइड्रोजन की जगह पे कुछ आर ग्रुप लगा दिया जाए, जिससे क्या है, बल्कीनेस बढ़ गई. एंजाइम को क्या पता, मूड बना दिया हमने उसे. So it will not be able to cut that amide bond. Okay, so people are working on macrocyclic peptides. अभी प्रोफेसर जैन से सर मैं मिला था. So he said I am now working with professor. It's a good thing that people are now, working as per the need. Peptide has to be delivered, but and more not only that, pharmacokinetic changes और भी बड़े एक्टिविटी भी बढ़ जाती है. अदरवाइज उसकी कन्फर्मेशन बहुत ज्यादा हो जाएगी. बेस्ट कन्फर्मेशन कौन सी है? एट द रिसेप्टर साइट विल बी डिफिकल्ट. सो एक और है मैक्रोसाइक्लिक मॉलिक्यूल्स. मैक्रोसाइक्लिक पेप्टाइड्स तो वन थिंग थी, एक मैक्रोसाइक्लिक मॉलिक्यूल्स बन रहे हैं, जैसे जैपेटोनिब, एलोटोनिब, एक एंकोलॉजी ड्रग्स हैं, उनके मैक्रोसाइक्ल्स आ गए. उनकी भी फार्माकोकाइनेटिक्स बहुत इंप्रूव हुई है और ड्रग फार्माकोडायनामिक्स भी इंप्रूव. सो दीज आर द न्यू पेप्टाइड्स. पेप्टाइड में चेंजेस मैंने बता दिए. मैक्रोसाइक्ल्स, प्रोटेक्स, एडीसीज भी आप साथ में लिख सकते हैं. ठीक है. चैलेंजेस क्या है? सी फॉर एवरी ड्रग ऐसा नहीं है कि आपने एक ड्रग ली, उसको मैं इसका चेंज करूंगा इसका हाइड्रोजन रिप्लेस करूंगा किसी और से. तो आप पहली शर्त that you should have an understanding of chemistry, you should be reading literature thoroughly. Second, optimize करना पड़ेगा. You need to standardize the reactions. It will take some time, maybe 10 दिन, maybe 1 month or maybe 2 months. You need to, but अगर वो हो गया तो think about that drug will give you so many new molecules without disturbing or without changing the route of synthesis. Already existing route, only one step synthesize करना last. ठीक है. फिर है expanding. यस, आपसे हमेशा पूछा जाता है केमिकल्स को, आपने नाम सुना होगा, केमिकल स्पेस बहुत बोलते हैं. फिर स्कोप ऑफ सिंथेसिस पूछते हैं जब भी कोई पेपर पब्लिश करते हैं. तो ये है कि आपने एक रिएक्शन कर दी, क्या वो रिएक्शन बाकी जगह भी एप्लीकेबल है? दैट इज कॉल्ड स्कोप ऑफ सिंथेसिस. फिर स्कोप ऑफ सबस्ट्रेट पूछते हैं कि आपने इस सबस्ट्रेट पे कर दी, क्या और सबस्ट्रेट पे भी? अब सैलिक ऑक्साइड पे हुई, क्या रोपिक ऑक्साइड पे भी कर सकते हैं? क्या हम इटोरिक ऑक्साइड पे भी कर सकते हैं? तो एज ए केमिस्ट पहले आपको उनका स्ट्रक्चर देखना है. अगर स्ट्रक्चर ऑलमोस्ट सिमिलर है, देन यू हैव टू थिंक अबाउट व्हाई इट इज हैपनिंग विद सैलिक ऑक्साइड, व्हाई नॉट इट इज हैपनिंग विद रोपिक ऑक्साइड. इफ द रिएक्शन गोस विद all the toxic, so that means your drug is, like you have developed a very good methodology. Okay, so sustainability, developing, greener तो मैंने आपको बताया, you have to think about the greener approaches. Okay, अच्छा, कुछ स्टूडेंट्स के लिए मैं लाया था कुछ ये फ्री केमिस्ट्री सर्च इंजन जो है. ये तो आपने थोड़े बहुत तो आपको पता ही है. कुछ नेम रिएक्शंस का आप देख लो, अगर आपको सर्च करना हो कुछ भी, ये सारे नेम रिएक्शंस ये सब पता है आपको. बाकी हम जो सर्च करते हैं जनरली हम एसटीएन, साइफाइंडर और जो बाकी सब ओपन सोर्सेज है, साइफाइंडर, एसटीएन है, उसकी रिएक्शंस समटाइम्स दे गिव एक्सेस. साइफाइंडर इज नॉट ओनली फॉर केमिस्ट, फॉर बायोलॉजिस्ट आल्सो, एनी गाय. Okay, so these are some of the subscription based chemistry search engines. Fortunately we do have at our university, Dr. Vishnu. We subscribe them. They are subscription. So these are इलेक्ट्रोसिस की बात की थी मैंने, जो इजे कोने टर्म दी थी कि पीछे से काटना. आपके दिमाग में मॉलिक्यूल है, मैं बनाऊंगा कैसे? तो कटिंग करना शुरू कर दो. रेट्रो मींस पीछे, तो पीछे से सिंथेसाइज करना शुरू कर दो. एंड थिंक अबाउट मैं कहां से रॉ मटेरियल लाऊंगा, कैसे बनाऊंगा. दैट इज कॉल्ड रेट्रोसिस. तो इसके लिए सारी वेबसाइट चाहे तो and note down. So thank you very much for patiently listening to me. Thank you so much. Any question? I would be happy to answer. Yes, sir. Yeah, actually, it is not that old. It started in 2005 or 6 onwards.
Okay.
one or two guys started working on it. Lekin now it is a fully established concept with various kind of catalysts which are costly, but no problem because at the end you are modifying them.
So does or hypothetically does that can lead to, better
Yes.
potency of the drug?
Yes, that you have to check it out.
Or lesser side effects of the drug?
Yes, why not? I gave one example. For example, in that molecule, if you have a free benzene or phenyl or phenylizine, so if it passes to the liver, it can get metabolized easily. Because at para position, generally hydroxylation takes it will be more water soluble. So in phase 2 conjugation, because the body wants that it should not stay in that it should clean out.
So are the drugs coming for CH?
Yes.
They're already in the market or?
Few are in market because people are still working on it.
Okay.
Not many.
Okay.
Late stage from science in the new concept.
Okay. Thank you.
Any question from audience side? Any curiosity question? No?
So they have understood everything.
Okay, if there is no question, thank you, Dr. Rajkumar, for discussing so many aspects of your advancement of synthetic methodology for drug discovery. You have discussed evolution of